Welcome, everybody. We have nearly 100 folks here, and I'm not going to reread titles because this really is a great collective we have before us. I'm just going to give introductions now with thanks to our speakers and those who are 100 of us in the room. Dr. Bill Gahl is an MD, PhD trained at MIT and the University of Wisconsin in medical genetics. He established the Undiagnosed Network at NIH, continues as its director and is a senior investigator of the medical genetics branch, as well as head of the human biochemical genetics section. He described the metabolic basis of at least 6 genetic conditions and has trained over 50 biochemical geneticists. Bill, thanks for being here. Dr. Ann O'Donnell-Luria is co-director of the Center for Mendelian Genetics at the Broad of MIT and Harvard, where she's an institute member, also an assistant professor of Harvard Medical School, member of the Division of Genetics and Genomics at Boston Children's, and associate member of the Center for Genomic Medicine at MGH, Mass General. Her laboratory within the Translational Genomics Group looks at large-scale genomic and transcriptomic approaches to increase the rate of rare disease diagnosis, and she sees patients with epigenomic conditions as a medical geneticist. David and Randie, I'm sorry, I think that's an L or an I, sorry, Randie. Harpell are the parents of a 20-year-old daughter named Kyla. Kyla did not receive a diagnosis for her rare condition until age 17, when she was diagnosed with Kyla NAA10 Ogden syndrome through participation in a rare Genomes Project research study. So we want to give all of our excellent speakers as much time as possible, and then we will come back for Q&A and discussion afterwards. So welcome again, and thanks for good active participation. I'm done.
Great, thank you. So thank you so much. And it's great to virtually be here to share some information about our efforts and along with Dr. Gahl's to improve diagnosis for patients. I am on several scientific advisory and clinical advisory boards that have no impact on the material I'll be talking about today, but just did want to share that. Okay, so genetic diagnosis is something that has become more routine over time, and yet 60% of people who go to clinic and have something like exome sequencing that looks at the spelling of all the genes, after that testing, 60% of people still do not get an answer. They don't have a pathogenic or likely pathogenic variant identified to say this is your genetic diagnosis. And to some extent, much of the management is clinical, and so you don't always need a genetic diagnosis, but we do think it's very helpful. And it's helpful because it tells you a lot about the condition. It can guide medical management, help you know what's coming, what things to watch out for, what screening to do. It can really assist in family planning by helping you understand what the chance of recurrence is in the family for the parents and also for siblings and other relatives. It can provide psychological relief. I think it's pretty common as a parent to worry about everything you did or didn't do. And there's always this like little voice in the back of the head of like, was it that hair product or could I have done something differently? And so understanding that there's a genetic cause and there's nothing one could have done or not done to prevent this gives a lot of psychological relief to a lot of families. Also, there's so much organization in the rare disease community, and some of that is around larger conditions, but some is very gene specific. And so building your network and understanding meeting other families who are going through similar situations can be very helpful. And so you need to know what the genetic cause is to identify that community and to participate in research. And as we're having hopefully an era where gene therapy and other more targeted therapies are really going to start to come into play, you're going to need a genetic diagnosis to be able to access those research opportunities and hopefully eventually clinical opportunities. So I wanted to learn a little bit more about all of you, who thank you so much for joining us today. We're going to launch a poll that's going to ask 3 questions. So has anyone in your family ever been in search of a genetic diagnosis? Yes or no? And then, so if yes, did they ever eventually receive a genetic diagnosis? And if your answer to the first one was no, answer no to the second one also. I'm realizing I should have had a not applicable choice, but forgot that. So answer no to the second if the first is no. And then third, how long was this diagnostic odyssey? And for some people, it may still be ongoing. How long have you or your people you know been looking for a genetic diagnosis? And we'll just take a minute here.
So we do have those results coming in. We have about 50% of people have answered. Let's give a little more time for additional responses.
You need some good Jeopardy music at this stage.
That's right. Just to add to the pressure of answering a poll. And it looks like participation slowing down. So I'm going to close the poll in 5, 4, 3, 2, 1. And here are those results. So you may see those to respond to, Dr. O'Donnell-Luria.
Yeah, so sorry, I was just making some notes too. So about half of people have been searching for a diagnosis at some point. 31%, again, didn't have the not applicable option here, but 31% said they eventually found a diagnosis, and others are still looking for answers. And then about half got the answer they were looking for in less than a year, or about 15% have taken over 10 years. And some of this is probably related to how technology and access has changed over time. But I think that's very, this is often a long journey to get a diagnosis. So great, thank you so much for sharing all of that. So one question is, why are patients remaining undiagnosed? Like, why are we unable to find a diagnosis for everyone? And one major challenge is access. Like, a lot of people, they either don't get referred to the right specialist to understand that genetic testing would be needed, or their primary care doctor doesn't realize genetic testing would be appropriate. And then for others, they try to send genetic testing or order genetic testing, but the insurance company won't approve it. And sometimes there's a sort of a lack of like knowledge and skill, like for the physician, that there's a lot of nuance to working with insurance companies to get them to cover it, and so they may not know that whole process or have time to go through the extensive rounds of appeals. And other times insurance companies just have a lot of policies against covering testing, and we do hope that that will change over time, but that's going to need policy changes. There are some things that may look genetic but may not actually be genetic. There are environmental or drug influences that can cause conditions that can mimic genetic conditions. An classic example was when thalidomide was prescribed decades ago now, and caused problems with limb development in fetuses. That was just due to something exogenous and not something genetic. There are analytic challenges, and I showed this Where's Waldo to give an idea of this. So we all have about 4 million variants across our whole genome if you sequence the whole genome. And so figuring out which of those variants is causing disease is a little bit like trying to find Waldo, but it's also like you don't necessarily know you're looking for Waldo, or you don't know what Waldo looks like. So it's a little bit harder than your typical, but just that there is a lot of variation, and the vast majority doesn't cause any health problems. And then there are technical challenges. So you might have not actually captured or represented the genetic change that's causing a problem in the testing strategy you did. And sometimes there's additional methodology you can do to pull that out of the data that exists. And other times you need a whole different technological method to identify the change that's causing a genetic condition. So it can be very complicated. So we started the Rare Genomes Project with the goal to increase access to genomic sequencing, given that, again, that a lot of people are unable to access proper genetic testing. Our goal is to identify causes of undiagnosed monogenic diseases, to share this information broadly to drive rare disease research, then to return results to families, but working in conjunction with their providers. And I'll go into that more actually, and then engaging participants as partners in this research process. And we do this with a large team, and it's very diverse expertise. So there's clinical geneticists like myself, there's many genetic counselors involved who really lead large aspects of this project. There are molecular geneticists and variant analysis who look at the genetic data and try and identify the variants that are potentially causing disease. This is vast amounts of data, and so we need a lot of technological and software solutions to improve our ability to go through all of this data in an efficient manner and find what we're looking for. And so we have software engineers and bioinformaticians on the team, along with statistical geneticists. So bringing all these expertise together we are able to identify genetic causes for a lot of families. And we also are a group that this isn't the only study we do. We also make resources for the community. We make one of the largest population databases called NOMAD, which is a representation of the genetic variation in the general population that's used in rare disease diagnosis by every laboratory. And we also contribute data to other resources like ClinVar that catalogs all the variants known to cause disease, and ClinGen, that's an expert group that provides a lot of guidance for interpreting genes and variants. So participants, to participate in the Rare Genomes Project, participants need to have or be a family member of a person with a suspected monogenic condition. This can be hard for families to know, but if a doctor has recommended genetic testing or been looking for a genetic cause, that means they're suspecting a genetic condition. Families need to reside in the United States, and that's because the regulations and laws that govern research vary country to country. And so our institutional review board that monitors our research is only able to do so within the laws and regulations of the US. Families need to have a medical provider. It can be a primary care provider or a specialist involved in their care, so that we have someone to partner with in returning the results and be part of that triangle with us and the provider and the patient. And then we tend to not work with, this study will not be effective or help families where the condition is not monogenic. So if multiple genes are involved, the study designed to identify those kind of things is different, and it won't work with what we're doing. There are some genes or some conditions where whole genome sequencing doesn't actually work very well, just because the genes are hard to sequence, and so we don't enroll those families because we know we wouldn't be able to find anything. And then we also do some funding restrictions initially, and also because there's a lot of other studies and expertise in this area don't work with strong family histories of cancer or isolated rare cancer phenotypes. So we recruit through a lot of methods. We partner with a number of advocacy groups. Clinicians a clinician referral is not required, but clinicians can tell families about the study or directly refer patients. We get a lot of people signing up through word of mouth, through social media, or one family sharing with other families about their participation in the project, and also through opportunities like this to share our work. Any family can sign up online. There's information they fill out there. If we do review the applications to see if we think they are likely to be monogenic or the study is likely to benefit the family. And then there's a remote video consent that can be a phone call if video is not an option. And we collect samples and pay for that sample collection along with medical records. We do genome sequencing and analyze the data. If we don't find anything, we do often employ other sequencing methods as we have resources. We're very collaborative and believe in sharing the data broadly, which I'll talk more about. And we return results that we think are contributing to the phenotype with the local provider, and discuss both with the local provider and with the patient participant potential next steps of like advocacy groups to join and other things. We have almost 1,000 families that have enrolled to date, with over 2,500 individuals from all 50 states, from the District of Columbia and Puerto Rico. They're actually about 85% of the US population lives in urban areas. So we have about a expected urban rural mix, very even male and female for the affected proband individual that's signing up. The individual with rare disease, we call that person the proband, the person who comes to medical attention. But they range in age from 1 month to 89 years. The 1 month old was actually a younger sibling of somebody already enrolled in the study, and that's how they got in so quickly. The 89-year-old was somebody who had been looking for an answer for decades for their genetic disease that I believe we're actually able to finally identify and give them that information. The study enrollment has been predominantly white, but there are some people from other ancestries or mixed ancestries or mixed races signing up for the study. This has been a major challenge. And one of the reasons we first developed this study was to try to decouple from people who have the resources to go to major academic centers. And so we are employing a number of strategies to improve our enrollment. So we have expanded to other languages, initially Spanish. I have 12 Spanish-speaking families that have enrolled to date, and actually just today got approval to start enrolling for other languages, and we'll be using phone interpreters for that. We also are trying things like mobile phlebotomy and other approaches to help families that are under-resourced and just don't have the time and bandwidth to participate in research, but have the interest to do so. So what are we finding? Well, there are about almost 600 families that have been analyzed. We have almost 140 results that have been returned. They're called reportable because some of them are still working their way through the process. Many of them are variants in known disease genes. And many of these people have had prior exome or other extensive testing. And it's just new information has evolved over time where we're able to do additional studies to resolve these variants and make a diagnosis. There are many new genes that have been discovered through this project, and again, in partnership with families. And then we work with these families and other science researchers and clinicians to contribute to medical literature so that these new gene discoveries are made public, and then diagnostic laboratories can start including them on the testing they offer. We also return strong candidates. We have again, our whole team meets and decides which candidates we think are strong enough to return. And so we actually have had 3 that after return were determined to be non-causal, and we think that's a reasonable number. We do want, especially if there's a test that the physician can do to rule in or rule out a diagnosis, we think that's very important to share this information. Moderate candidates are generally not strong enough to return, and most of these will actually not turn out to be the diagnosis. And then over half of families remain undiagnosed still. And we're not done here. We're still trying other approaches. But this just, again, highlights that even with whole genome sequencing all the bases, there's bases of DNA in the genome, there's still a lot of challenges here in interpretation. So just to very briefly go through what we're finding, we find new disease genes through something called genomic matchmaking using the Matchmaker Exchange Network, where we submit a gene and a candidate along with the high-level phenotype information and connect with other researchers and start to build these patterns, and are able to, again, put together enough patients and enough medical information to understand that there's a new gene here. We spend a lot of time trying to resolve variants of uncertain significance, so rare variants that we don't understand the medical impact of. And we do that by identifying more patients that have the same variants, and again, comparing the phenotypes and the patterns we see. We also use something called RNA-seq that looks at the expression of a gene. And if you have a variant that impacts expression or how the gene is processed, that can also be an important clue that the variant is a disease-causing variant. And then we have some ability, or to build further functional studies through collaborations. And so we work with a lot of groups around the world to try to really take these rare variants and see what else we can learn about them. We've also done a lot of work with small structural variants or tandem repeat expansions, 2 types of structural variant. I'm just showing you one example here. This gene normally should be read from left to right. And so, it starts here and you go through and read the gene. But what has happened is there's an inversion. So this first part of the gene is broken off and flipped around. And now when you read the gene, you start reading here, and you don't ever, you go to the left and you read it backwards. And so you don't ever see this other half of the gene. And if you just did exome sequencing, it just sequences the little boxes. And so you would never understand that the gene is not put together right, because all the pieces are there, but they're not in the right order. So those are the kinds of things we've been able to find. And we share the data pretty broadly but in a protected way. The data is de-identified, but it is genome data. There's phenotype data that's shared too, and it's put into a repository that's developed by the National Institute of Health called Anvil, where researchers who have studies that have permission from their institutions to use genomic data can apply for access to this data and apply new methods to see if they have other ways that they can diagnose patients. So rather than my telling you specific stories about individuals we were able to diagnose, we thought it would be much more helpful and meaningful to actually invite Kyla's family to speak with you today. So Randie and David Harpell are here to share information about their experience for looking for a diagnosis.
Good afternoon. To introduce ourselves, we're Randie and David Harpell. We live in Wall, New Jersey. We have been married 27 years. I turned 35 years old in August 2001. In January 2002, a positive pregnancy test confirmed that we were expecting our second baby. Due to my age, it was labeled a high-risk pregnancy. When we got to the doctor's office, however, she told us that she knew we were pregnant as there was a sac, but there wasn't a baby. She sent us home telling us that it was probably too early. Come back in a week and we'll check again. And true to her word, the next week we were able to see the sac and the baby. We also went to a specialist who took measurements and thought that the baby looked fine. And then the rest of the pregnancy was uneventful. On August 29th, 2002, 3 weeks before I was due, Kyla Ricky entered the world. She did have a slight fever and she was taken to the NICU for observation. Because she was 7 pounds, 14 ounces, the physicians decided that they would not consider her preemie. She was pretty fussy. Her coloring seemed a dark reddish color. And that's next, if you want to change it. So the picture on the left there shows that her coloring at that point. She never wanted to be put down and would nurse a lot. I started noticing that Kyla would not look at me when she nursed. So we took her to the pediatrician to ask his opinion. He sent us to Children's Hospital of Philadelphia to see an eye specialist. And at that point, she got her first diagnosis, delayed visual maturation, meaning that she was basically blind, but eventually her vision would kick in. Then my sister-in-law came down in November. She's a nurse practitioner, and she felt that Kyla was floppy with low muscle tone, so we called an early intervention to come, and they were coming on New Year's Eve. I actually did call them to cancel because I wanted to give the spot to a more needy child. But then I learned that Kyla qualified in every area for early intervention. And then she got her second diagnosis of hypotonia. At that point, she started therapies. I went back to work part-time as a teacher for the rest of the school year, and all my days off, we had appointments with various specialists. The neurologist then gave her a diagnosis of hypotonic cerebral palsy, a term they use for children with global developmental delays. Then we had some scares. We were rushed by our neurologist one night to Children's Hospital of Philadelphia for muscular concerns. Then her soft spot didn't close when it was supposed to. The soft spot on her head. So it seemed like everything eventually would kick in, but at Kyla's own pace. I began reading her the book, Leo the Late Bloomer, and I realized she was my Leo. The good news was that the specialist couldn't find a reason why she was so delayed, but the bad news was we didn't get a diagnosis. So Kyla became Kyla, developing at her own pace. She talked and walked much later than the typical milestones. We went through genetics at Children's Hospital of Philadelphia at that point, but got no results. She was 3 years old, in the bottom right picture, when she went to the preschool handicap program. The school bus picked up this little girl who was in diapers, who didn't talk, who wasn't really aware of the world. And then we as a family started to adapt to everyday life. We pushed as many therapies as we could on Kyla. We celebrated every achievement, but it quickly became apparent that although she was in a public school preschool handicap program, she still needed more than it could provide. We provided private speech and physical therapy after school for a time. As you can imagine, with 2 working parents and an older sibling, coordinating all these services were a challenge. Once Kyla graduated from the preschool program, she entered a multiply disabled program for kindergarten. You could change the slide. The placement was perfect for Kyla. She had some very tough teachers who pushed her to her limits with tough love. They made sure she was accepted by all the other students in the school. It was as if she were a celebrity. Everywhere we went, people would say, "Hi, Kyla," and even though she wasn't too aware, and because she wasn't too aware, she wouldn't be able to tell us who they were. Fast forward about 4 years. Kyla was going through the motions of all aspects of life. She developed into a happy girl, laughing all the time and developing a great sense of humor. She, during these 4 years, was given a diagnosis of cognitively impaired. She was given a diagnosis of PDD, pervasive developmental disorder, but only because we wanted to try ABA therapy with her, and an autistic diagnosis would allow insurance to cover it. Kyla continued to puzzle all the doctors. She was able to read. She taught herself at about 3 years old. She could decode words that she had no comprehension of. She has a calendar in her head and relies on routines. She spoke in words, finally, that the family began to understand. Fine motor and abstract thinking, math included, continued to baffle her. I relaxed with attempting to diagnose her. But at that point, I joined a group on Facebook called SWAN, Syndromes Without a Name. Many parents would list their children's symptoms, asking if their child had those characteristics, and if so, what was their diagnosis. Other parents posted that their children were finally diagnosed with whatever, and these are the symptoms that their children were showing. I searched a lot to no avail. Then in September 2013, Kyla, at 11 years old, began what we thought were asthmatic symptoms. She had difficulty breathing. I called the ambulance twice. One time she was blue. Eventually, in November, at Children's Hospital of Philly, we found out she had T-cell ALL leukemia. Again, it was a very rare diagnosis for girls and the harder of the leukemias to treat. After a 2.5-year grueling treatment, we are happy now to say she's a warrior, a survivor. This year will be 10 years. In 2014, the oncologist asked us if we wanted to get genetics evolved again to see if something was related to the leukemia. Again, we didn't get an answer. Nothing popped up in her genetic workup. You could change the slide. Then around 2017, someone posted on the SWAN Facebook page about a rare genomes project out of MIT and Harvard. At that point, Dave and I decided we would try to see if we can get a diagnosis. Like Anne said before, I always felt like I had Jewish guilt. Was it something I ate, something I drank that could have been the cause of her difficulties? Would there ever be a way to cure it that I wasn't aware of? I began working with Grace from the Rare Genomes Project, and a few months later, she told us we were a good fit for the program, and she wanted blood from Dave and I to see what was different from Kyla's and our genetic makeup. We didn't hear anything until about a year later when I got a letter saying they have a diagnosis. Grace got in touch with us and our pediatrician and finally diagnosed her with NAA10 related syndrome, Ogden syndrome. And you could change it. Ogden syndrome is basically a genetic mutation on the NAA10 gene. Grace spent a lot of time explaining this to us. Kyla's case is de novo, unique to her, not to Dave and I. We found out the reason she wasn't diagnosed earlier was because the researcher hadn't discovered it until 2009 and published it until 2011. This was 2019. Kyla was 17 years old. At that time, there was only 56 girls identified. Now there are over 100 children diagnosed from all over the world. Grace next put us in touch with the researcher. His name is Dr. Golson-Lyon. He explained the syndrome to us and he gave us some resources. He has told us that many of the children with Ogden syndrome do not walk or talk. Some have seizures, some have heart issues, neither of which Kyla has luckily, but is something for us to continue to watch out for. Dr. Lyon is still researching and finding that there's a wide range of abilities. And Kyla, being as unique as she is still the only child identified with the specific variant that she has. We always say she's 1 in a million. We then joined the NAA10 Ogden Syndrome Foundation. There's a website, a Twitter, and an Instagram, which provides information about NAA10. The group also petitioned Washington, D.C. to list NAA10 as one of the recognizable syndromes. And just this week, they posted an 8-minute video to raise awareness of Ogden syndrome, which is now on their website. You could change. Fast forward to 2023. Kyla is thriving in an out-of-district placement where the emphasis is on functional skills. She has begun learning various jobs. She's working on her communication skills and enjoying seeing her friends. I retired after 33 years of being a teacher. I felt that when I retired, I wanted a new purpose in life. So I began to get involved in the Ogden Syndrome Foundation, which meets often with Dr. Lyon. Dr. Lyon's working on a cure to fix the NAA10 mutation. He's developing a genetic cure by attempting to overexpress a normal copy of the gene NAA10. They're doing this by using something called an adeno-associated virus, which I practiced saying last night, which is just a way to introduce this gene into many tissues of the human body. They may also try a different approach using something called lentivirus, but that will depend on funding. Last summer, I began assisting Dr. Lyon with research. I had the privilege of interviewing the other parents of children with the NAA mutation from all over the world. What I found from talking to everybody is that maybe I can't change Kyle's development at this point, but maybe I can help the path of the parents who are just starting out with their little ones. They have received a diagnosis at a young age. They have a large support group already in place. They have been told what resources are out there for their children. Basically, a lot of PTOT and speech. So now we finally have a diagnosis, a name, but it doesn't come with very much guidance. All we really know about Kyla is what we learned by being her parents. I know we were always on the right path with her with OT, PT, and speech. Kyla's a perfect human being, a young adult who teaches the world to be more accepting of people with special needs. Dave and I are very blessed to be her parents, but that doesn't mean we don't have challenges. We do have many challenges that we're working to overcome. Her life is totally in our hands, and some days it is exhausting. We worry for her future. If something happens to us, what will become of her? That uncertainty is my largest worry. She will never be able to live independently. I count my blessings that Kyla can walk and she can communicate. She has a community of genetic sisters that she meets with on Zoom calls. She has a place she is completely understood and accepted. Who could ask for anything more? Back to you.
Thank you so much for sharing, Randie. And we'll hopefully have some time for questions at the end. I just very briefly wanted to share how you can get involved. If you already have, again, we're talking about genetic diagnosis here. So if you or somebody you know has a genetic diagnosis, there are ways to share your genetic test report through Genome Connect. And then they gather that information to make sure that everyone in the community knows that that variant is associated with disease, so it can help in others getting genetic testing. And they also register your interest in the variant. So if anything, new information comes that changes the interpretation of the variant, you would be notified. So you can check that out at their website there. And for anyone who's still searching for a diagnosis, we're still enrolling families in the Rare Genomes Project. And I invite you to check out more information on our website where you can apply directly, and our email address is also there in case you have questions. So I wanted to, again, hear from the group if for those who have faced obstacles or heard of other obstacles or just sort of interested in this space, what do you think are the biggest obstacles to receiving a diagnosis? And we've talked about some of these today, like insurance, access from insurance, getting to the right doctor through a subspecialist access. variants of uncertain significance and understanding if these are really causing disease or not, or just not having testing being negative and not being able to find an answer. Those are some options. There's many other obstacles. So we're gonna, I think, launch a link. There should be a link in the chat that will have a place that you can go and enter up to 3 of obstacles that you faced or you think are some of the biggest in this field. And then we'll share the results from that. So we'll take a few minutes now.
As Dr. O'Donnell Luria mentioned, that link is there in chat. That'll open up a separate browser window where you can enter some responses and we see the ones coming in.
Variant interpretation, a lot of challenges there's so much variation, and really figuring out how to understand it and which variants are going to contribute to disease is a major challenge. It's moving around a lot. Some issues with costs of testing, lack of access to services with long wait times. Motivation, there's so many other things to focus on, to find the time to continue to push forward to try to find a genetic diagnosis, not knowing how helpful it's going to be or not for you or your family. Functional testing and figuring out, and our hope is that over time, like there might be some access to functional testing through the diagnostic laboratories for at least certain tests, but otherwise it's connecting with researchers and putting research effort into looking into specific variants, which can often be like a huge amount of work. And then actually knowing if that functional test is really how well it works for a condition is also tricky, just because there's many things you can show an altered function, but it doesn't actually mean it causes disease in a human. And also I see doctors not believing, like it is tricky when there's so many, often multiple medical problems, and kind of understanding how this comes together and being able to like communicate this with a healthcare provider that is able to listen appropriately and work with you through this process can be challenging to find. Just to make sure we have enough time for Dr. Gahl, I don't know if it stays open or not, but thank you for sharing all of this. And I think we should probably move on to the next part.
Thank you. Yeah, that word cloud will continue to accept responses even as we transition here to Dr. Gahl's presentation. And I'm going to share this now. Please put your questions in chat as we go. There will be time at the end of this presentation for Q&A here, before then gathering back in Zoom 1 for cross-pollination with the other session.
Well, thank you very much for inviting me to this New York Mid-Atlantic Caribbean Regional Genetics Network meeting. And I want to add special thanks to Joanne Badertha for inviting me for this. Today, I want to talk about the NIH Undiagnosed Diseases Program and how it moved to a national program and then an international program. I have nothing to disclose, but I do want to disclose acknowledgments to the director of our Pediatrics Undiagnosed Disease Program, Cynthia Tift, our director of bioinformatics, David Adams, our head neurologist, Camilo Toro, and our pediatric neurologist, Maria Acosta, and also support from my institute, NHGRI, also the Office of Rare Diseases, and the Clinical Center of the NIH, and the NIH Common Fund that has supported the network for several years now. In addition, there have been many contributors on the staff of the Clinical Center and some of the individual institutes here at the NIH. In addition, incredibly help from the patients and their families who have contributed so much. Well, the UDP was started in 2008 to help patients who have undiagnosed diseases and have had difficulty reaching a diagnosis. But we also have to discover new cell biology and biochemistry and physiology and genetics. So that is our scientific goal as well. And the way this works is that applicants submit their medical records along with the referring physician's letter. And then I triage the patients for the adults and Dr. Tiff for the children. And we ask our intramural consultants to review the records and decide if this is a good case for us. We make the final disposition and send a letter. And we really accept about now maybe 1/3 of all the cases that apply. But those that are accepted are seen as inpatients at the clinical center, which is the largest research hospital in the country, for a 1-week stay. And there's no charge to the patients. So we've seen over 5,000 medical records and admitted and evaluated over 1,500 individuals. About 40% are kids and more than 50% have neurological diseases. We've done a lot of exomes and genomes, made about 400 diagnoses, and have about 200 publications. The genetics that we do is personalized, depending upon the phenotype. And we do commercial testing when it's indicated. We do SNP array analyses, and we do exomes and genomes often on the families. But the phenotyping is one of the most important issues here. And we're able to get a lot done in a week because we don't have to ask for insurance payments or approval. We also do functional studies on some of the variants that we find in the next generation sequencing. So I'm just going to give you one example of discovery of a new disease. We had an 18-month-old female who had failure to thrive. She also needed total parenteral nutrition. And her main finding was hypopigmentation of her skin and hair with poor visual acuity, along with organomegaly. Her liver and spleen and kidney were both enlarged and had storage material in them. She also had hypotonia with developmental delay, but she had no osteopetrosis. And this is going to become important in a few slides down the pike. So here's the young lady, and you can see how hypopigmented she is. You can see her, let's just put this thing on here for the laser pointer. You can see her hypomelination as well. And here is the accumulation of storage material within the liver, histiocytes, within the duodenum, but there are cellular inclusions here and here. And these white things here are in the PMN. And in fibroblasts, she has vacuoles as well, which you can see with some staining here as well. Then we found a second patient who was from Ghana, also had the hypopigmentation, the organomegaly, the storage, the delayed myelination, and again, no osteopetrosis. And this is the child right here, hypopigmented compared to his dad, with hypomelination as well. And the inclusions here, you can see this type of thing, and this in a lymphocyte. And again, the fibroblasts show a very similar pattern. Both children, it turns out, on next generation sequencing, had de novo mutation in CLCN7, the exact same de novo mutation. And CLCN7 is a chloride transporter that's important in acidification of the lysosome and also the endosome. So when a proton pump pumps protons into a lysosome, eventually there's an electrochemical gradient that forms on the inside membrane of that lysosome. And then the next proton that tries to get in has difficulty because of that gradient. That gradient needs to be dissipated by a counterion. And that counterion is chloride. And the transporter for that counterion, chloride, is CLCN7. So if you don't have enough CLCN7, in other words, loss of function, you're not going to be able to acidify your lysosome. And acidification of the lysosome is incredibly important in the breakdown of bone by osteoclasts. So there's a known disease associated with CLCN7, biallelic loss of function variants, which are actually these mutations all along here. And that disease is osteopetrosis because you can't break down your bone properly. This is different. this is a different mutation, a specific one right here. And that suggests that this is a gain of function. In other words, too much chloride going in, too much acidification. So Joseph Mendel in his laboratory in NINDS put the CLCN7 into xenopus oocytes and measured the current across the xenopus oocyte membrane. And when he put in the normal, he got this much current. When he put in the mutant, he got this much current. So an increased amount of transport, and that translated into having an increased amount of acidity or a decreased pH in proband 1 and proband 2, measured in fibroblasts using Oregon green fluorescence measurement. And you can see the pH is lower here than in normal and lower in this proband as well than the normal. So this is hyperacidification. And in fact, it's dominant. And you can tell it's dominant because you take normal fibroblasts and you transfect them with the mutant CLCN7, and you get those vacuoles. So even though there's normal, two copies of the normal CLCN7, if you put in the mutant, this is what you get. Well, we created a mouse model, Ralu Nicolai and May Malakdan did, and the model is hypopigmented. And the fibroblasts from the model have the storage granules, and the liver pathology also has storage granules, which you can see right here. So it recapitulates the particular disorder. Well, it turns out that if you have too much acidity in a lysosome, you can alkalinize that with chloroquine. And as you increase the concentration of chloroquine, you decrease the amount of lysotracker red staining, lysotracker red being an indication of acidity. In addition, the increase in chloroquine treatment in concentration right here increases the pH, meaning it alkalinizes the lysosomes. Well, this brought up the possibility to Dr. Deborah Day Salvatore in New Brunswick, New Jersey, who is the physician of the second patient that I showed you, the possibility that you could treat with chloroquine. She did after getting IRB approval, and this decreased the patient's kidney size on ultrasound, and the patient was able to do things physically that he could not previously do. However, there was enough damage done that both of these children eventually succumbed. On the other hand, there are new patients that we've identified who have slightly milder disease who may be amenable to being treated with chloroquine. And there are a couple of groups that are also working on allele-specific oligonucleotides to try to knock down the gain of function in the mutant in this individual, in individuals who have this disorder. Well, that was one example of a new disease discovery associated with a unique phenotype. But the Undiagnosed Disease Program has done that about 24 times, actually a few more that aren't on this list, associating a particular phenotype, which is reasonably unique, in fact, very unique, with a particular disease gene. So those are discoveries, but in addition, we've made a lot of diagnoses. And I'm just going to show you some of them here. These are lists, and some of these on these lists you may consider should have been diagnosed, but these patients were at major medical centers throughout the country without a diagnosis, and sometimes it's because the phenotype is a little bit different, and our examinations expand the phenotype, and sometimes it's a new disease. Sometimes it's really, really unusual. Like at the time, there were only 5 in the world with SCARB2 mutations. Now there are a lot more. And you can see here 20 families for this disorder, etc. C9orf72 used to be a very unusual disease. Now it's a relatively common diagnosis to make, and some very unusual diagnoses here as well. Actually, I have 12 slides of this, but I'm only showing 6 because this gets sort of boring and redundant. Some recent diagnoses here, etc. In addition, we've made 10 diagnoses of lysosomal storage diseases in adults who had attenuated disease because it was late onset. Now I want to mention a few examples of personalized medicines. In other words, times in which we were able to treat a patient because we knew the defect, or sometimes even when we didn't, but largely when we did. We had a 65-year-old man who had recurrent meningitis. And of course, meningitis is very scary because when it's bacterial, it can progress rapidly to death. When it's viral, it can last longer, but it's still a very serious disorder. Well, this individual had probably of the order of 20 or 25 episodes between the ages of, let's say, 58 and 65. And several of them caused him to go to the emergency room and get a spinal tap, where a pleocytosis was found. So he had white cells indicating inflammation in his cerebrospinal fluid. An exome indicated that he had a particular mutation in NLRP3, which was known to be associated with Muckle-Wells syndrome, an autoinflammatory disorder, and was known to be heterozygous and gain of function. So we knew that NLRP3 had a complex, which was a group of proteins that made up the NLRP3 inflammasome. And the function of this was to convert pro-IL-1 beta to active IL-1 beta. And the one component that he had a mutation in was always on. So in other words, it was a gain of function, so that there was repeated episodes of too much conversion of pro-IL-1 beta to the active form. We reasoned that we could treat with an IL-1 receptor inhibitor, anakinra, and did that. And within 4 hours, this individual, who was plagued by a bad headache and by inability to walk and concentrate, and was confined to a wheelchair at times in an episodic fashion, in 4 hours, he was able to walk and talk normally. So that was directed therapy. Here's another example. We had a 22-year-old woman who had dystonia. So when she was a child, she gripped her pen improperly, and the teachers, of course, told her, you got to stop doing that. But the reason she did that was because the muscles of her fingers were pushing her fingers around the pen in that fashion, and she couldn't constrain them. She also had an impaired gait. But the biggest problem for her was with her tongue, because she had involuntary tongue movements, which impaired her speech, impaired her swallowing, and she couldn't eat properly and went down to about 80 to 85 pounds as an adult. She had monoallelic KMT2B mutations before we knew that this was associated with dystonia. This encodes a histone lysine methyltransferase. Well, we put her mutation along with a list of other mutations, made it public. And Dr. Kurian, who runs a dystonia clinic in London, called me up one day and said, we saw that gene on your list. We have in our clinic 20 patients who have mutations in KMT2B with dystonia, and 5 of them have responded to deep brain stimulation. So she published this, and then within a couple of years, we saw another patient, Dr. Ariana Soldatis and Dr. Camilo Toro, saw a 20-year-old with progressive dystonia who had roughly the same history as the patient that I just mentioned. So foot drag, impaired gait, needed a wheelchair for long distances, was on baclofen, couldn't write properly because of her fingers again, etc. And I'm going to show you some film of her. Here she is. Let's see. If this works. This is the pen, though. And maybe here. Okay. So I'm going to just take this off. So here she is in bed, almost apostatnic. She has normal cognition, able to answer questions fine.
Consuming.
And she points.
What's your favorite Taylor Swift song?
She can't speak properly because of the dystonia.
You Belong to Me? Is that it?
Sing here with.
No, I don't.
Her favorite Taylor Swift song is You Belong to Me.
Oh, is it With Me? You Belong to Me.
With-
That makes more sense.
And what's your other favorite?
What's your favorite?
Well, we did a workup, did a lot of things, didn't find anything except she had a de novo KMT2B mutation here. And that meant that we could suggest deep brain stimulation. And she received deep brain stimulation. And after that, she was able to walk. And in a minute, you'll see her walk up steps. I think we need to emphasize that when you have a person who's really debilitated, that what you might consider, a small advance like being able to walk is actually an incredible advance because of the ability to care for oneself and to do things independently. It's incredibly important for the patient, but also for the family. And of course, we see a lot of individuals who are in such a debilitated state. So what did we learn? That KMT2B is a relatively common cause of dystonia, that the oral motor problem is really prominent, that deep brain stimulation helps, but maybe most importantly, that sharing is incredibly important in order to develop treatments and understanding of these rare diseases. The final case I want to mention is a 14-year-old female from Nigeria whom we never saw, but were able to offer some advice for. At age 1, she had knocked knees and couldn't walk properly. She had growth delay, and then a bunch of fractures where you're not supposed to ordinarily get fractures without trauma. And her diagnosis was rickets. So they were treating her with surgery and vitamin D. You can see the growth arrest lines here, the fractures, etc. And here she is, casted a lot. Well, her diagnosis was clinical rickets, and that can be due to vitamin D deficiency or hypophosphatemia, maybe because of renal tubular Fanconi syndrome and hyperphosphaturia, or metabolic acidosis. She had low serum phosphorus and mildly elevated alkaline phosphatase, which is a response of bone breakdown, low serum bicarbonate and potassium, but normal vitamin D. We found a de novo pathogenic variant in SLC4A1. And this is a chloride bicarbonate exchanger in the distal renal tubule. So she had metabolic acidosis as the cause of her rickets. And the treatment is alkali with either bicarbonate or citrate plus potassium. So that was, and Carlos Ferreira and Ellen McNamara worked on that case. Our program also trains postdoctoral fellows and post-bac ERDOs, who are individuals who've just gone to college and they're planning to go to medical school or graduate school. Our trainees have been first authors of 26 undiagnosed disease program articles. We have a short course that Tom Markello taught. He also taught this at Berkeley and UC Santa Cruz and NIEHS. We have a journal club, weekly patient rounds, where now I see about 60 or 70 individuals in the audience as participants, young people, and we present cases every week. We have genetics fellows rotating and guests from around the world. This, for example, is one of our older pictures of the young people who are listening to case presentations weekly. Made a lot of publications, including in some high-impact journals, given a lot of talks around the world. Have Congress people come occasionally and get a lot of congressional inquiries, a lot of lectures and press. And the press was really important because it allowed us to expand to a national network in 2014 with 6 extramural clinical sites, a coordinating center, some sequencing cores, metabolomics core, model organisms core, a screening center, etc., and a central NHGRI IRB protocol with reliance agreements with the other centers, formal data sharing agreements, and the patients, by their consent, agree to sharing of personally identified information within the network and de-identified information with investigators who are collaborators to try to find their defect. So here are some of the places that were part of the first phase of this. And then it was expanded in 2018 to 11 extramural sites with the addition of a few, but it retained the coordinating centers and cores. And we're continuing to work on sustaining this. So the network, over the course of roughly 7 years, saw over 6,000 applicants and evaluated over 2,000, made over 600 diagnoses, and did a lot of exomes and genomes. Well, in 2014, this is expanded by virtue of the fact that there are now undiagnosed disease programs, not just around the country here. I'm going to do this again. Sorry. Not just around the country, but around the world. These were undiagnosed disease programs there. And the Undiagnosed Disease Network International was founded in 2014 and has now had actually 11 international meetings with its own charter and committees and data sharing policies. And some of the recent initiatives are to have a website for data sharing, including cases that are presented by the Diagnostics Working Group, which was just established. And we've established a Developing Nations Working Group as well, which has representation throughout the world. You can see here on this map. And the Developing Nations Working Group, in concert with an advocacy group for undiagnosed diseases called the Wilhelm Foundation, has established a champions program to identify physicians in 4 developing countries. We're going to expand this later, but those countries are Congo, Ghana, Pakistan, and Mali, where those champions can collaborate with members of the Undiagnosed Disease Network International for consultations, for sequencing and training. In fact, there's training in Olaf Bodemer's lab at Harvard for such individuals. And this can serve as a clearinghouse for the givers and the receivers to work together and collaborate. In addition, there's a philanthropy that can be involved in this to essentially expand undiagnosed disease programs to make them worldwide. This Champions Initiative has really stellar clinical ambassadors in low and middle income countries and is supported by experts in the UDNI and also by the Wilhelm Foundation. So we're hoping that the undiagnosed disease programs will expand throughout the world to promote the United Nations initiative that no person with a rare disease is left behind, and to improve the quality of life for individuals with rare and undiagnosed diseases throughout the world. I think any of you can work with an undiagnosed disease program, in particular ours at the NIH if you want, by referring undiagnosed disease patients to us. But also we have a database called UDPICS that has genotypes and phenotypes in it. And if you have a particular gene of interest, you can ask us, in particular sending me an email, and we will search and tell you if we have a patient with a variant. We'll give you the list of variants in that gene, and you can decide if you're interested in them. And if you are, we can tell you in a de-identified fashion what the phenotype is, and you can decide how you want to proceed with that. We can also collaborate with you on research into new diseases, if you like. Our goal is to lend a hand to those individuals who suffer without a diagnosis and who eventually, most often, have a very rare or a new disease. Thank you very much for your attention. I think I can take some questions a little later.
Alyssa, how do you want to handle our time?
Well, we do need to head over to the main session momentarily, but I want to give you guys a chance to ask Dr. Gahl or Dr. O'Donnell-Luria or Dave Randie a quick question if you have anything to put in the chat now.
Yeah, I put a connecting question, so let's see if
Yeah.
somebody else has a question specific here. Great talks, lots of different approaches, appreciate sort of the realities of, you know, getting out to people, getting the word out, as well as particularly to the Harpells, you know, getting additionally involved now in the patient potential connection sort of things, because we had talks earlier on how difficult it was for lots of limitations getting to appointments, all the rest of those things, as well as the QI talk that was going on in parallel here.
Yes, thank you again for sharing your story. So what we're going to do together now, if there's no questions at this exact moment, is follow the link in the chat back to the main session, and you'll have a chance over there to share your kind of key takeaways. And if a question occurs to you as you're in cyberspace getting over there, you can ask the question over there. So everybody come follow us back to Zoom 1, and we can do it there. Thanks, guys.